Today I approach the topic as an ecologist of soil communities. I will highlight some of my research that demonstrates use of compost to suppress soil-borne pathogens, as well as introduce new tools for testing suppression. Not only is composting a solution for organic waste management and soil fertility, but it has an added value as a tool for plant growth promotion and suppression of plant diseases. Throughout this talk, I'll be referring to what we call thermophilic compost, that is material that has achieved the temperature and turning requirements for pathogen reduction. The least diversity of soil communities in the compost are at the peak thermophilic stage. After this, it becomes repopulated with beneficial or biocontrol organisms, which are referred to as antagonists on this slide. Curing and maturation are essential to the type of disease suppression that I'm referring. There are a number of serious soil-borne pathogens in the horticulture industry. Most of these are genera that have global distribution and a wide host range, causing serious economic losses. This slide shows a few examples of diseases that can be managed by disease suppression, and more complete coverage is in the book chapter listed at the bottom of the slide. Just to highlight a couple, we know that Pythium and Phytophthora are both classified as Mycota or water molds. Their biology is greatly affected by irrigation practices, and past research indicates that it's possible to have a relatively good control of these pathogens with compost by managing the compost recipe to give a competitive favor to the biocontrol over the pathogen. Rhizoctonia, on the other hand, has a different ecology. It's a strong saprophyte, meaning it survives on detritus between the plantings. And the historic use of compost to naturally suppress rhizoctonia has been less successful, more in the order of 20%. Illustrated is historic work by Harry Hoyntig, who clearly demonstrated that feedstock quality really alters the competition between the pathogen and biological control. Illustrated are disease incidence data for a disease called Fusarium wilt of flax. At that time, the horticulture industry was highly dependent on peat moss as the potting media, but it was very conducive to the disease. This led to development of a compost recipe containing hardwood bark. Hardwood bark contains complex chemicals that are antagonistic to the pathogen. Now, if this compost was fresh and young, there was some disease suppression, but it was much more effective if the material was allowed to mature. So one generalization we can make is that most reliable disease suppressive composts are those made with woody materials like tree bark, wood chips, and woody yard trimmings. I pose a general hypothesis. The pathogens are copiotrophs, meaning they have a sweet tooth. What do I mean by that? I mean they do very well under high nutrient conditions. This is especially true for Pythium and Phytophthora that are poor competitors that make them reliant on high nutrient conditions to thrive. In contrast, the Rhizoctonia are more competitive because they can also metabolize cellulose and starches, which are found in hay and leaves. And it takes a mature compost containing the hardwood bark to provide complex carbons that then favor a biological control community that can truly antagonize pathogens. In recipe development, all we need to do is really change the source of carbon to completely change the bacterial community composition. There are distinct patterns of microbial communities in mature compost that contains common source of manure mixed with different sources of carbon, as illustrated on the left. On the right is illustrated how we can alter the bacterial community also simply by changing the type of curing process, whether it be windrow, aerated static pile, or vermicompost. So with the exception of Pythium and Phytophthora diseases, different compost recipes and maturity affect pathogens and host crops differently. Plant bioassays are considered the gold standard for measures of suppression, but they are quite laborious. Therefore, simpler and rapid indicators have been used to assess the quality of compost. We define compost as, quote, stable when microbial activity has subsided because they have completed their degradation of organic wastes into a stable form. Respiration is a measure of activity. However, it can be difficult to interpret. For example, in the event that a material has become anaerobic or extra dry, this can artificially reduce respiration. Diversity indices are simple to compute, but reveal basically nothing about who is in the community or how or what those community members are doing. These simple measures have been reasonably good to predict Pythium ultimum and Pythium irregulare, but not Rhizoctonia. Furthermore, the methods are criticized for their imprecision and the weak associations with populations of known biological control agents. I want to spend the remainder of my talk on tools that have predictive power of plant bioassays but are quicker and provide more ecological insight as to who in the consortium is there and what they are doing. What do I mean by consortia? They are communities of microbes working in concert to perform specific ecological functions. A simple measure is a ratio of fungi to bacteria. Greater values of this index suggest there is greater maturity. The phospholipid fatty acid, the PLFA method, is a cost-effective way to obtain data for a ratio of fungi to bacteria and has become one of the most popular methods for measuring microbial biomass and broad community structure. However, there is some debate over the taxonomic groupings, and the method is not very sensitive to compost types. Compost plate assays can be used to quantify how much of the indigenous microbes in compost directly inhibit the pathogen, and this can be completed within 2 to 3 days. For each sample, we pour a compost slurry into a pair of flasks containing molten agar. One flask contains living microbes and the other is autoclaved to kill the microbes. Each of these is then poured into petri dishes that solidify and a plug of the pathogen is added. The suppressive colonies create a visible zone of inhibition around the pathogen colony, and we quantify the suppression as the difference in how the pathogen grows on the plate with living microbes from the pathogen on a plate with dead microbes, and this is computed as a rate per day. This indicator consistently differentiates among the curing process, the maturity, as well as the recipe of compost. Here is evidence of the application of the plate competition tool. The term raw refers to living microbes. The term autoclave refers to the dead microbes or sterile conditions. Here's clear evidence that the pathogen suppression is based on living microbes. We can see also that the suppression is greater in the vermicompost than that with hardwood bark. Ecoenzymes are another tool that focus on the type of carbon and nitrogen status of the substrate. Microbes produce suites of ecoenzymes to acquire energy or nutrients when they become limiting. The tool can be customized to any substrate of interest. It is a colorimetric method performed in a microplate and read by a plate reader, and one can process dozens of samples within a day. Ecoenzymes are able to track the temporal change in bacterial and fungal communities throughout the composting process and are a relatively promising indicator of disease suppressive compost. Simple carbon compounds such as starch or cellulose degrade first, with complex carbon compounds such as lignin degrade later. Ecoenzymes active on cellulose and chitin are better predictors of disease suppressiveness by fungal pathogens than microbial respiration. The cell walls of Pythium and Phytophthora contain cellulose. The production of cellulase damages those walls and weakens the pathogen. Similarly, abundant rhizosphere bacteria producing chitinase, which is the constituent of cell walls of fungal pathogens such as Rhizoctonia, Fusarium, and Verticillium. Commercial kits come pre-populated with carbon and nitrogen substrates where you can add a compost slurry and the wells change color and the intensity of that color can be quantified by spectroscopy. This color represents how actively that particular substrate is being consumed by the microbial community in the compost. This approach is useful in screening among compost recipes and communities of relative suppression. We used this assay to identify functional species. A functional species would be one that uses a unique combination of the 31 carbon substrates. Here we identified 29 functional species of actinobacteria. The actinobacteria are known to be suppressive to pathogens. We used it to test the method of anaerobic soil disinfestation, and we can see how this community of actinobacteria shifted from one community composition before and to another one after the treatment. Going back to my hypothesis about pathogens as copiotrophs, our tools indicate that poultry manure and litter-based compost are conducive to pathogens such as Rhizoctonia solani and E. coli. We know this is not just simply ammonium content, but is actually microbial in nature. We want to learn who is there in that consortia that's responsible for the conduciveness. The difference we're seeing are in these bacteria, the sphingobacteria, the flavobacteria, that are 2 to 4 times more abundant in poultry litter compost than they are in the dairy manure compost. These bacteria are members of the consortium or superorganism that support copiotrophic organisms to acquire carbon, and sometimes we just label them as cheaters. We need to learn the pivotal points to intentionally design compost to suppress disease. Illustrated is how addition of compost changes the fungal community of soil and how that community is distributed spatially. The center of each pie are phyla, and the outer ring is taxonomic class. You can understand the graphic simply by comparing colors and changes in the color. First, unamended soil has a baseline community, which is changed by the addition of vermicompost. Adding the plant changes the unamended soil more than the one with vermicompost, which seems to have a buffering capacity. The community again changes once a pathogen is introduced, and there becomes a spatial difference in the community in the soil surrounding the root compared to the inside the root. The pathogen is inside the root, and you can see it represents a fairly large portion of the root community. In conclusion, emerging tools are effective in predicting disease suppression. The community approach is more effective and consistent than working with one or a few populations or isolates. And different pathogens on different crops require different consortia, which leads to the suggestion of creating designer composts. So for future research, we know that the development of consistent products with disease suppressive properties demand a better understanding of ecology and their mechanisms, so we get the right players there. There are no current regulations or guidelines that define the desirable microbiological properties of compost. However, with a better understanding, we can start to learn the pivotal points where they can be managed to enhance disease suppressiveness. It's quite likely we'll need to develop designer composts that are specific to different diseases. I cannot do all this work alone, and I wanted to introduce you to the team behind the scenes conducting these experiments at University of Vermont. Thank you.